what I'd like to talk about is global health. And yesterday there was a very good introduction by Trevor Mundell. And I would like to dig a little deeper and first of all share with you one of the challenges that we have. So the three major diseases in global health are malaria, as Trevor mentioned yesterday, tuberculosis, and HIV. And with tuberculosis and HIV, the intersection between them is particularly dramatic, and I'm going to come back to that in a moment. So about half the world's population is at risk for malaria. One child in Africa actually dies every 60 seconds from malaria. And malaria costs Africa more than $12 billion a year in lost productivity and in GMP. So these diseases, actually not only are they devastating from the perspective of health, they're also devastating from the perspective of the economy. Tuberculosis, a third of the entire world's population is latently infected with TB, over 2 billion people. That's an astounding number, particularly given the fact that we don't know what latency is. And I think Sebastian pointed out very nicely that most of the dogma that we have in the TB field is 100 years old and is wrong. And the consequences of not knowing anything about these diseases can be horrifying, particularly when you consider this huge amount of latently infected people with TB. I might add, I too am latently infected. I grew up in Africa. 2 million TB-related deaths per year. And as Sebastian mentioned, we have obviously lots of drug-resistant TB, but in South Africa at the moment, in India and in Iran, total drug-resistant TB has been found. And that is obviously also pretty scary stuff. HIV claimed more than 40 million lives, and 1.5 million people a year die from HIV. There have been an enormous number of kids orphaned by HIV AIDS. And I was in a village in Kenya once where the oldest person in the village was a 14-year-old girl. So besides the death and destruction, can you imagine growing up in a place where the oldest person, the caregiver, is 14 years old, and the social ramifications of that? Now, no vaccine exists for any of these diseases. Drug resistance is rampant, and as Sebastian pointed out, diagnostics are 100 years old. And obviously, all of this leads to major problems. So what I'd like to share with you today is the following. First of all, as I learned when we started the ISB from Lee, partnerships are absolutely critical in doing this kind of work. And secondly, that you always collaborate with the very best people. And those are two things that have stuck with me ever since. So what I'd like to do is, first of all, discuss how I and others have set up 2 institutes in Africa, which will serve as field sites and build capacity in Africa at the same time. I'd also then like to discuss this issue of diagnostics within TB, and I'd like to show you our recent work in which we now, I believe, are able, based on blood tests, to predict the progression from latent to active TB. And this is not a trivial finding, because if a third of the world is infected latently with TB, you really better be able to tell who's going to go from latency to activity. And then finally, I want to show you our efforts on using systems biology to accelerate the development of vaccines. So let's start with the partnerships. So there are 2 reasons why we focused our efforts on South Africa. I come from South Africa originally, and I have a political past there. And because of that, the government, I've built up a lot of trust with the government. And so that was one place where we could easily get access. The second reason was the infrastructure, which I'm going to come back to now. But the third one is that South Africa has the dubious honor of having the highest incidence of HIV worldwide. So if you look at sub-Saharan Africa, HIV has claimed more than 40 million lives there. 350 million people are living with HIV over there, but they say 25 million is higher. 1.5 million people per year die over there, and 2.4 million people are newly infected with HIV. So if you're going to study this human disease and you're going to study it in people, which is the only place you really can study it, you better be there. Secondly, TB in South Africa. 80% of South Africa's black population is latently infected with TB, an astounding number. TB is the leading cause of death in South Africa. We've discussed the drug resistance within, Sebastian discussed it within the rest of the world as well as in South Africa. As I mentioned, we now have totally drug-resistant TB, and TB is the leading killer of people who are HIV infected. Because HIV, we know that when the immune system drops, HIV the immune system drops, latent TB is activated, and HIV obviously takes out the immune system. So Nelson Mandela, who was the first president of Free South Africa, basically made, I think, a very cogent point. We cannot win the battle against AIDS if we do not also fight TB. TB is too often a death sentence for people with AIDS. And I think that was extremely perceptive at the time that he said it. Sadly, his successor didn't believe that HIV caused AIDS, and that caused us a lot of problems in the field. Okay, but South Africa, I believe, will be the battleground for the eradication of TB and HIV for reasons I've mentioned. It also has a very strong infrastructure. It has need to make a progress here, and that is, I've mentioned the reason for it. It has the political will to do so. It has very strong public health expertise, and I'm going to come back to that. And it has a very, very strong clinical trial and research expertise as well. So this is definitely the place to go. So many years ago now, well, I guess it was, I was still at the ISB, a few of us went to the HHMI, made these arguments, and said, if you want to impact this disease, give us the money and we can start an institute within South Africa. And we thought we'd started in Cape Town where the best science is, but we made a decision, we'll put it in Durban, which is on the East Coast, which is where so much of the TB and HIV is. In other words, in the epicenter of the pandemic. And so a lot of work later, we've managed to set up the institute. This is our beautiful building located in the center of the Nelson Mandela Medical School in Durban. The main funder of this was HHMI, who put more than $100 million into the project. And we've established very, very good relationships between this institute, which is called KRIT, Kwazulu-Natal Research Institute for TB and HIV. And it's become, I think, quite a force in TB research worldwide. The NIH, that's Fauci, has also seen this as an important area for study, and they've supported us as well. And many people have been involved. I still want to see Lee Hood here with a mask on. That is his good friend, David Baltimore. So they have a friendly competition all the years, so Lee, you better be there, otherwise David's got something up on you. This just shows David also knows more about wine than Lee does, but that's another story I won't tell you about. Anyway, within KRIT, this just shows some of the technologies, the absolute best stuff you can buy with HHMI behind us. We managed to do this. This year is a remarkable microscopy setup. It's within the BL3 facility, and this year is Alex Segal, who did his PhD at the Weizmann Institute with Uri Alon, and he did his postdoc with David Baltimore. and he is now a young faculty member at KRIF. So the point is we've been bringing in really, really promising young people into the institution, and they do a couple things. First of all, I think they're doing cutting-edge research, but secondly, we're very much interested in capacity development. These are young kids that come in, etc. Final thing I'd like to say about our containment facilities, which can contain total drug-resistant TB. We have laser capture microdissection there. We have a CORS system there, which it does metabolomics and so forth and so on. Now, I'd like to show you one project, and only one, which is ongoing, and this is the only slide you're going to see, touching on this issue of latency. Now, the thought has always been that latent TB are within the granuloma in the lung. I myself have doubts about that, but anyway, this is the current dogma as well. And it's kept in check by the immune system, because if you get HIV, it activates, and even if you give Enbrel, which is a drug that made Immunex worth $16 billion when it was taken over by Amgen, it reverses or it antagonizes TNF, tumor necrosis factor, it reactivates. But there's no good animal model for this. We need human tissue. And since I mentioned that 80% of people in the area have a latent TB, Sebastian mentioned the cavitation and lung resection, they resect lungs all the time. We get access to those lungs. We can pull out the granulomas using laser capture microdissection within the BL3, and then study the cells and the bugs, etc. Recently, Gary Nolan, who's been a close colleague for many, many years, has joined the fray with his MIBI technology, which he discussed yesterday. Now, we also had established another institute, which we called the Africa Center. And this one was in population health. And it also looked at TB and HIV, and it looked at it, though, in the context of essentially public health, epidemiology, et cetera, we followed these people in this area, who are 200,000 people, of which 87% of them have latent TB, and 42% are HIV positive. I mean, these numbers are just staggering. This is the area of Somkele. It's situated over here on the east coast of South Africa, and this here is Durban, where KRIF is. So it's about an hour drive between the two places. So we have this unbelievable treasure trove of information, which was very well done over a 16-year period in the Somkele area. And so now we had the idea to put together the Africa Center with KRIF, so that we would have cutting-edge science, but at the same time have field sites where all of this, epidemiological and public health data existed. So within Somkele site in KwaZulu-Natal, this is the problem. First of all, the lifetime HIV acquisition risk for a 15-year-old girl is now 80%. Astounding. You get more drug-resistant TB over here than in the US and UK combined. So it's a very significant amount. And finally, current prevalence within the antenatal clinics, women 20 to 40 years old, 68% of them are HIV positive. So like I said before, this is ground zero. If you're going to make an impact on the disease, this is where you got to be. And so this is the Africa Center. It's got field workers, got map-based homestead identification, a lot of GPS work in here, et cetera. I won't go through all these details. We've kept blood spots for 16 years. That's the building over there. And there's a lot of demographic information, et cetera. I won't go into this. Wellcome Trust from Britain has funded this entire institute. And Wellcome Trust also is the exclusive funder of the Sanger Center in Cambridge, and they are now sequencing 50,000 genomes for us, full genomes, out of the 200,000 people that exist there. And we know who will be sequencing, and we've spoken with Lee, with Gustavo, and others about how to do this properly. So this is what we now have. We've called it ARI for Africa Health Research Institute. We've got the Kairuth site in Durban. We've got the Somkele site. And we've gone around, and I've gone hat in hand and collected now, we've got, I think, 165 million, basically from Wellcome Trust, from HHMI, from NIH, from the Gates Foundation, which is what Trevor was referring to yesterday, the Max Planck Institute, and also from the South African government. Okay, and this is ARI. So we also have industrial partners, most important of which are Novartis, GSK, and Sanofi. So now what I'd like to do is go to science. And firstly, to tell you about our work in predicting and understanding the progression from latent to active TB. So TB, again, it starts with somebody who's infected. They essentially cough, and either they get cured or you get a latent infection. We've discussed that. 10% of those will go to active TB, and of those, worldwide, 2 million die. So we decided, based on something that we had decided, this, Lee, I pulled out from the first retreat that we had in Friday Harbor Labs for the, and look, it's still the old style slide. Anyway, we discussed there using leukocytes as sentinel cells. And the thought there was they circulate through all the tissues, therefore they patrol the body. They interface and participate with infection. They're abundant and can be readily purified. Multi-parameter analysis can be done, and expression patterns give you molecular signatures, which you can, and this can result in appropriate therapy. So even then, we were thinking, think about many of the areas that we're discussing today. So the first study I want to show you about took place in South Africa as well. Now, this one is on the west coast in Worcester, South Africa. This is what the place looks like. And this was done in collaboration with SATV, which is the South African TB Vaccine Initiative, led by Willem Hanekom. And what we did there was we wanted to see whether we could predict which MTB-infected adolescents would progress to TB. So we took 6,500 of them, and we just did blood draws on them every 6 months for a number of years. And then we came back and looked at the whole blood transcriptome, and we profiled the transcriptome, the RNA, and then we looked to see whether we could come up with a signature that would be predictive. And indeed, we were able to do that. I won't obviously take you through this, but suffice to say that this signature is part of a type 1 interferon response. And that came as a big surprise to us, because the interferon response is normally associated with viral infections, and yet this is a bacterium. And there are very interesting aspects to that observation that we're following up, and I won't have time to go into. But what I would like you to look at is the progressors are shown in purple, the non-progressors are shown in green. I just want to take you through the signature, and if this thing goes purple, we correct that this is predicting transition to activity. So this is a year and a half before diagnosis. This is a year before diagnosis, and this is 6 months before diagnosis. So this particular network is highly predictive of who's going to go from latency to activity. Moreover, when people become active, they're obviously put on drugs. And if those drugs work, which mostly they do, you can see that the network is relaxed. So this is not only a predictive diagnostic for who's going to go from latent to activity, but it also allows us to stratify people at various levels. In other words, are they relaxing their signature or not? What do we do about that, etc. So this has just been published in The Lancet. First author there is Dan Zak, who's a postdoc in my lab, and he's now a faculty member over at the Center for Infectious Disease Research. And we were actually able, while a coup was basically taking place in Africa, we were able to displace them off the front page of the most read newspaper in South Africa. All right. Now, how can we take this and get some functional data out of it? In other words, this is a signature, but it doesn't really matter what it is. The question is, now we want to go and say, what do these nodes do? And are they actually functionally related to the transition? And two postdocs in the lab, Tom Horn and Pierre-Yves Bachou, who is, I'm sure, well known to you, had been doing candidate genetic studies in a number of diseases. What we were doing was we were identifying lots of innate immune molecules, and then we'd look for associations between mutations in those innate molecules and various diseases. So basically a candidate approach rather than GWAS, which didn't exist at that time. So we found a whole lot of associations. I won't go into them, but these are the ones that are associated with MTB. And so many of these markers that are diagnostic also are associated with a variety of TB phenotypes. And so that gave us more confidence that there was a functional connection. Now, one fascinating thing is that there's a TOL2 mutation which is associated with TB meningitis. So people with that particular mutation get meningeal TB, but not pulmonary TB. And so this is giving us a lot deeper insight. Okay, so now I want to take you to vaccine development. And I'd like to tell you what I think can be done here. And I'm going to focus on HIV vaccines for the moment. So I think that systems biology can allow us to identify signatures for vaccine immunogenicity and protection. It can allow us to evaluate vaccine trials, which are really critical because they cost a lot of money and they take a long time. If we could understand the molecular networks leading to a protective response, I believe we can re-engineer the human immune system to give you a protective response. And what we've been focusing on there are synthetic biology approaches in the main. So let's look at, I'll give you a vignette of each of these. So let's look at the identification of signatures for vaccine immunogenicity and protection. So the study I want to tell you about is one with SIV. Now, obviously, we cannot infect people with HIV. So we have to go to the macaque monkey and infect those monkeys with SIV, which is the simian form of HIV. And the question we asked was, can innate gene expression predict the adaptive immune phenotype? We don't get cure yet, but can we predict the adaptive response, a correct one? Moreover, what we can do is look at the viral load. And the hypothesis here, and may or may not be correct, but I think it is if there is a higher viral load, then you've got less of a protective response. If there's a lower viral load, you're doing better. And that is the outcome that we looked at. So this is the study. We took the candidate vaccines and adjuvants, put them into the macaque, looked at the innate immune response, which takes place in hours and days, and we did those from blood cells and from lymph node biopsies. We looked at the adaptive response, which looks at weeks and months. Here we were looking at CD4, CD8s, etc., we've discussed today as well. Then we challenged after months and looked at the SIV replication rate. All of this is computationally integrated, and then we try to find innate networks that were predictive of immunogenicity and protection. And as we always do in systems biology, it's an iterative process. We then go back to the beginning and do it all over again, hoping to get better and better. So what we wanted to understand was, can we predict from innate signatures, which occur in the first 2 weeks, what the protection or the immunogenicity against HIV would look like a year hence when they've been infected? Because if you could do that, you could test whether a vaccine is working or not very early on. The STEP trial vaccine took almost 10 years to do. It was a miserable failure. I'm going to talk about it in a moment. And if we had known within the first weeks or months that it was going to fail, we could have pulled the whole thing, we could have retooled and done it again, okay? So there are consequences to this. So I won't take you into this. This is from a lymph node. Obviously, as always, stuff goes up, stuff goes down. But we then did some quadratic discriminant analysis where we wanted to fit a model that would predict the appropriate CD4 response based on the expression of pairs of genes. So we're doing it's a machine learning algorithm, basically. We're looking at pairwise comparison and just saying any two transcripts, if we compare them together, are they predicting a low CD4 response appropriate for the virus or a high CD4 response? And what you can see here is that in 56 monkeys, so this is a huge trial, a very expensive one actually, you can see that in general, and for the sort of aficionados, this was constrained with an elastic net penalty, etc. We heard far better talk than I could ever give yesterday from Sushi. So just to look here though, is that red on red, this is pretty damn good actually. There are a couple of outliers, but, and you can't see this, there are lots of green dots on the green. So this actually allowed us to begin to see whether we were getting the right immunogenicity for the virus. Another study that we did, as I mentioned, was we wanted to see what the effect was on high and low SIV loads after infection. This is a year later. And based on the transcriptomes from the innate time, which is 2 weeks about, you can see again using this pairwise approach, again we could predict low SIV or high SIV pretty accurately, although there were some outliers as well. So this was a network of signatures which expressed CD4 responses, which suggested CD4 responses and the SIV load, taken after 2 weeks and looked at a year hence. And the edges here, the blue edges, are gene pairs that are associated with enhanced CD4 responses, and the red edges here are gene pairs associated with decreased viral load. And we were able to correctly classify the animals to the extent of 83%. Now, this sounds impressive until you think about it. A coin toss would give you 50%. So it's not as impressive as you want it to be. So we started adding additional parameters, hoping to get a better idea of a better predictive idea. And so here, diamonds of information from the lymph nodes, circles are from blood cells. Yellow was on day 2, orange was on day 6. Around the circle here is positive correlation. If it's red, if it's green, it's a negative correlation. So the more parameters we were adding, we actually got much better. So here, we got greater than 90% predictive accuracy. And that starts taking you into the realm where you're prepared to say, well, I'll do something. However, this was in monkeys. So how can we do this in people, which is obviously our main? So we went to the Somkele site. And there you get people that are HIV progressors, which means that once they're infected, they very rapidly will progress to full-blown AIDS. On the other hand, we also get controllers. These people get infected, and it takes a long time for them to progress to AIDS. And we made an assumption. We said the reason they are taking a long time to progress has got something to do with the immune cells, with the immune responses within the body. Whether that's true or not right now, I'm actually not 100% sure, but I think it is. And so we looked at the HIV-specific immune cells from progressors and from controllers, developed networks, and actually it's beginning to look pretty good, although we're not there yet. So the next step I'd like to do is show you how we evaluated vaccine trials. So this was the STEP trial from Merck. So Merck came up with a Gag-Pal-Nef vaccine, and they put it into adenovirus as the vector. And this thing took almost 10 years. I don't want to even tell you how much money this cost. It makes my good friend Roger Perlmutter at Merck, who runs the place now, essentially gray and almost bald. So here, what it was, you took the cold virus, adenovirus, And within that, you put the plasmid, GAG, pol, nef, and then essentially this was put into the host and antigen presentation and so on. And this was the step trial result, okay? And so this is the vaccine here, and this is the placebo, and this is HIV on the left. And so first of all, the horrifying result after all this time was the damn thing doesn't work. All right, terrible. Worse yet, okay, didn't end there. We found that people with pre-existing antibodies to the viral vector were actually doing much worse. And this is a problem. So here we got the people on the placebo. This is the viral load here. What you can see is that the people with the vaccine had more viral load than the people on the placebo. This was devastating because not only was it obviously a disaster from the perspective of making a vaccine, moreover, it was a political disaster because one of the sensitivities about going into Africa is that you're not practicing on people over there. You are actually bringing something of value. And so this was a really difficult thing. Now, so this raised the question, if you have pre-existing antibodies to the vector, why is it that you are getting more ill? And I'll just give you a hint about this. We've been doing this in the lab. It turns out, I think, that when the vaccine vector is opsonized with immunoglobulin, it goes into the antigen-presenting cell through the FC receptor, as opposed to the way that the virus would enter the antigen-presenting cell another way. Since it's in a different compartment, I think it's presented differently, and this is why you get the result. But nonetheless, if we had that information up front, we could have stopped, I think, the trial. So we had, at the same time as the STEP trial was going on, together with Julie McElroth at the Fred Hutch, we were doing the same trial. We were using the Merck vaccine. We were looking at, here in Seattle, at men that have sex with men, so that's a high-risk group. We were running a vaccine trial concurrently. And basically doing profiling, as is still the days of microarrays, of PBMCs after primary vaccination and so forth. And also we did some immunological responses. And obviously we started developing networks for the Merck Ad5 vaccine. And this is what we found in the end. And this is really stunning. And Anaya talked about some of these signaling pathways earlier today. But this is the RIG-I network. It's a virus detection network. And what you can see here is that the entire network is activated when people have Ad5 negative titer, meaning absent the vaccine, they're doing better because their bloody viral detection system is better. But when people are Ad5 positive, then they essentially are down-regulating their viral detection system. Same thing here with the inflammasome network, which Naya talked about earlier. If you have been exposed to flu virus, which is the vector, and it's opsonized, the inflammasome network, which has an antiviral component to it as well, is downregulated. And the toll network, which, as you know, also is a detector for infections, the same thing. So this was absolutely, I think, stunning result. And basically what it said is the virus-responsive innate immune networks appear to be attenuated in the Ad5 people, and one of the reasons it happens is because their antiviral defenses are down-regulated under those conditions. Much remains to be done, and we'll get to that another time. So the next thing is we wanted to understand the molecular networks leading to a protective response. Now, I'm not going to show you a lot of experiments here because it'll take us a long time, but suffice to say that over many years now, we've been looking at macrophages and dendritic cells. Those are the two cells that present antigens and essentially orchestrate the ensuing immune response. And these are some of the papers that we've published over the years. And if you're interested, you could go and take a look at those. But here, for example, you have a network which is stimulated by the toll pathway, and it's a pretty complex network. This is in the interferon pathway, which, if you recall, is associated with TB. And so the idea here, and we've done a lot of motif scanning, chip transcriptomics, we've done epigenetic profiling, and as I mentioned, we've done genotyping. So take all this information together and see whether or not we can get an association of these pathways with a protective response, okay? And so, and we're still obviously struggling with this, but we are, I think, making good progress. In addition to the transcriptional networks, we also wanted to look at the signaling networks. And this work was done by together with Rudy Ebersold and Rob Moritz. And here we were looking at the phosphorylation networks, the signaling networks within these cells. Now, if you take bone marrow-derived macrophages and you stimulate them with a Toll-4 agonist, you find about 15,000 phosphorylation sites in about 4,000 proteins. And 7,000 of those phosphosites have never been reported before. Now, bear in mind, I've got tenure at Rockefeller, okay, for looking at one kinase and one substrate. So this is pretty damn rich for people who need jobs down the road. Anyway, we could also look at the kinase cascades by inference and also by phosphorylation events, and also we were able to identify kinases by a biotin, globally by a biotin assay. So in addition to this, we did lipidomic profiling, also in a lung disease. This was influenza. And here what we did was bronchoalveolar lavages, and we looked at the eicosanoid profiles because they are molecules that turn on and turn off inflammation. And we have the capacity to, in a large scale and global way, measure 140 of these metabolites. And you see this here, and that was published a couple of years ago as well. So, so the question is, if we have the networks that are essentially result in a protective response, can we essentially reprogram the human immune system to respond effectively to these diseases for which there seems to be no vaccine or cure? And so this we're busy with now. So we've got a lot of networks from dendritic cells, which Jim Heath explained yesterday, presents antigens to T cells. And now we are taking normal synthetic biology approaches and using them to essentially see if we can perturb these networks to drive the networks themselves to essentially instruct the dendritic cell to evoke a protective response in the T cells. Obviously, if we can do that, then we could look for small molecules which could do that as well. And then if they are added to vaccine formulations, hopefully we can make some progress there. The last slide is looks at some of the real challenges we are confronted with still. And I think that we're confronted with this in the entire area of health going forward. First of all, we have differential effects of vaccines. So BCG, which is the only kind of vaccine that you have for TB, works some places, doesn't work most places. So why is that? We have which I'm not talking about at all, a malaria vaccine, which doesn't work in Mali, although it seems to work elsewhere. Why is that? We need to be able to stratify these places so we can essentially tailor the vaccines for the right place. We haven't even touched age in the immune response here. We know that kids have a different immune system to adults who have a different immune system to the aged. Kids are the ones we vaccinate. So if we want to vaccinate adults down the road, you know, are we actually even making the right vaccines? And then, of course, there are the genetic differences. Do we need to develop different types of vaccines for different genetics? So just to acknowledge very quickly the people that have done the work: Mark Gilchrist, Kate Kennedy, Liz Gold, Alan Dirks, Garnett, Steve Ramsey, Peter Askovich, Becky, and the work was done, the TB work was done by Dan Zach in the lab, who is now a faculty member across the street as well, and he was working with these people. The TB study was done together with SATVI, the South African TB Initiative. Willem Hanekom led that. The Merck trial was done together with Julie McElroth at the Hutch, and the monkey stuff was done with Louis Picker at OHU and with Bob Cedar at the NIH. And finally, much of this has been done together with, at least the thinking of it as well, has been done together with John Aitchison, who seems very keen to get back on the stage and throw me off, but I have to acknowledge that as well.
Thank you, Alan.